Hello everyone, welcome to our first lecture in chapter 11. We're going to take a brief detour away from nervous tissue and the nervous system for a moment, and we're going to cover part of the muscular system. We're going to be covering muscle tissue. We're going to be looking at how muscles function, and then after this we go back and we will spend the rest of the semester looking at the nervous system again. So let's start with our not really attendance questions. First, what does it mean if a protein has quaternary structure? And next, what is an epimysium? So go ahead and try to answer those before listening to the correct answers. And let's see what those answers actually are. This first question goes way back all the way to chapter 2 at the beginning of the semester. What does it mean if a protein has quaternary structure? Well, remember when we were making protein, we have the sequence of amino acids, that was the primary structure, and then based on those amino acids and their R groups, they kind of coiled up or folded up into alpha helix or beta pleated sheets. That was the secondary structure. And then those regions interacted with each other and it folded up and took on a three-dimensional shape called the tertiary structure, and at that point we had a protein, and it was that protein shape that dictated what it did, and we said every protein had a tertiary structure. But some proteins took it even further. Some proteins, we would have tertiary structured proteins connecting with other tertiary structured proteins to make a bigger protein complex, and that was quaternary structure. And next, what is an epimysium? This comes from the early self-study part that was actually at the start of chapter 10, which goes along with what we're doing right now. When we looked at muscle, if we looked at a whole muscle, there was a membrane of connective tissue that surrounded that whole muscle, the muscle organ, and that surrounding membrane was the epimysium. Now, there's quite a bit of self-study in this chapter, so make sure that you are doing that because all of that material is fair game for an exam. I did try to, as always, keep the self-study more anatomy related and isn't really tough material. We're going to spend lecture talking about the physiology of muscle tissue. So let's start by talking about some characteristics of muscle tissue, and some of these will probably sound familiar because they share some characteristics with nervous tissue that we just looked at. For example, first, excitability. Remember we said nervous tissue had excitability, well so does muscle tissue. Really all cells have excitability, that just means they respond to stimuli. But nervous tissue, and here muscle tissue, take that to the extreme. They are very responsive to stimuli, and the stimuli are going to cause them to have very, very specialized responses. Next, also shared with nervous tissue, and that is conductivity. Well, the excitability in nerve and muscle causes an action potential. It causes electrical activity. And then that electrical activity is conducted along the length of the cell. We saw that in neurons. We're going to see it again in muscle cells. Next, contractility. This is the very, very specialized thing that muscle cells can do. Contractility means that in response to those electrical signals, muscle cells are going to be able to shorten. So that is the contractility. Next, extensibility. That means that we can take muscle cells and stretch them a little bit. Obviously, we can't stretch them indefinitely. They do run the risk of tearing, but muscle cells are really good at being stretched. And finally, muscle has elasticity. That means if it is stretched, it will snap back to essentially its resting length. So all of those things are characteristics of muscle tissue. But really what we're going to be spending this lecture on is looking at the microanatomy of skeletal muscle. And even though I just said we weren't really going to be looking at anatomy in the lecture, that it was going to be self-study, the microanatomy is important for us to spend time in lecture on, because the next two lectures, when we're looking at how muscle functions, it's going to really depend on this microanatomy. So I want to spend time in lecture talking about this. So what we see here is a muscle cell. And like we saw with nerve cells, muscle cells also look really, really different from that generalized cell that we looked at back in chapter 3. They are very specialized, and because they are very specialized, they have a very specialized shape. And like we saw in the nerve chapter, we're going to give some very different names to some of the organelles and regions of a cell that we have already learned. So, the muscle cell itself, which we see right here, and we have part of it cut away so that we can see the inside of it, a muscle cell will have a few different names that we can give it. We could just call it a muscle cell, but we could also call it a muscle fiber. Those two terms are interchangeable, muscle cell, muscle fiber. And we could also call it a myocyte. M-Y-O, myo, is a root word that means muscle. So we're going to see that a few times in this chapter. So muscle cell, muscle fiber, myocyte. They all mean the same thing, and that is this big thing that we're seeing right here. Now, muscle cells are pretty different in that they are really long. When we talk about cells, generally they are so small that they are microscopic, of course. We saw them through the microscope in lab. And muscle cells, their diameter is also microscopic, but they are really, really long. A muscle cell runs the entire length of the muscle organ. So, for example, in your upper arm, we have the biceps brachii, the muscle that we generally just call a bicep. But the biceps brachii, it runs from about your shoulder to about your elbow. Well, the muscle cells in the biceps brachii also run from about your shoulder to about your elbow. Now let's look at some of the regions and some of the organelles that we see here. This yellow sheet-like structure, this is in any other cell what we would call the plasma membrane or the cell membrane. In a muscle cell, we're going to give it a special name. We're going to call it a sarcolemma. Sarcolemma. And a sarcolemma is just the plasma membrane or the cell membrane of a muscle cell. But it has some unique features that we don't see in other cells. Here and there, there's these holes, or they look like holes, but in reality, they're not. They are infoldings of the plasma membrane called transverse tubules, or T-tubules. T-tubules are spots along the plasma membrane where the membrane folds inward and it forms a tunnel. It's these purple guys right here. It forms a tunnel that goes through the entire muscle cell and out the other side. But it's not a straight tunnel. It doesn't go straight through. It actually branches and forms these long, twisting passages. So those are transverse tubules, or T-tubules, and they are just extensions of the plasma membrane, or of the sarcolemma. So if we were to go inside of one of these transverse tubules, we would be completely surrounded by plasma membrane, by sarcolemma. Next, inside the cell. Remember in inside of a cell, everything other than the nucleus, we called the cytoplasm in a typical cell. Well, in a muscle cell, everything inside besides the nucleus is called the sarcoplasm. So sarcoplasm is just the cytoplasm of a skeletal muscle cell or of a muscle cell. Now, I just mentioned something that I want to talk about here. It's not in our notes, but I said everything besides the nucleus. Well, the vast majority of the cells in our body have a nucleus. But muscle cells, some of them, skeletal muscle cells at least, which is what we're talking about in this chapter, we're going to see at the very, very end there's some other types, but skeletal muscle cells actually have more than one nucleus. We say they are multinucleated. So one skeletal muscle cell will actually have a lot of nuclei. Now, when we look at this picture, we could see these long structures here. These are called myofibrils. And myofibrils are these long strands of several protein bundles. So it's a lot of protein strands joined together in bundles that run the entire length of this muscle cell. That's called a myofibril, and there are several myofibrils in this muscle cell. Also, here and there, and I don't see it shown in this image, but there are clusters of glucose kind of clustered together. You remember we called that glycogen. We learned about that back in chapter 2. When we have a lot of glucose, we string it together, and it's called glycogen at that point. Well, in muscle, we find chunks of glycogen called glycosomes. Glycosomes are just clusters of glycogen. And remember, glycogen is just a lot of glucose joined together in these chains. Well, why would we want that? That's because muscle is actually really ATP dependent. We use a lot of ATP in muscle. And when you're making ATP, remember what we need? We needed glucose. Well, instead of waiting for the body to deliver glucose, muscle carries its own. It has glycogen stores in the form of glycosomes. And along those same lines, muscle has something called myoglobin. Myoglobin is a protein very closely related to another protein called hemoglobin. You've probably heard of hemoglobin. Hemoglobin is what's in our blood that makes it red. And hemoglobin is really good at holding on to oxygen. Well, myoglobin is almost the exact same thing. It's found in muscle instead of blood, and it holds on to oxygen. It's what makes muscles red. When you go to the store and you buy some meat and it's red, that's because of myoglobin. Well, really, it's because they inject it with some red food coloring to keep it red, but meat is red because of myoglobin. Why would we want myoglobin in the muscle cells? Because just like we saw with glycosomes, we need oxygen for ATP production. So the muscle cells have myoglobin to keep a local supply of oxygen. Both the glycosomes and the myoglobin are found in muscle so that this muscle has a ready supply of the ingredients needed for cellular respiration. And we can see in this picture, muscle cells have a lot of mitochondria. These mitochondria draw glucose from the glycosomes and oxygen from the myoglobin to make ATP. Next, another organelle that we learned about back in chapter 3 was the smooth and rough endoplasmic reticulum. Well, the smooth endoplasmic reticulum of skeletal muscle is very, very modified. It branches and gets really, really thin and really, really thick throughout the entire muscle. So since it's so modified in muscle, we give it a special name. We call it the sarcoplasmic reticulum, or the SR. The SR, the sarcoplasmic reticulum, is just a smooth endoplasmic reticulum. And it has these really, really thin areas that branch throughout the muscle cell. And then it has these very, very enlarged, swollen areas that we see right here, this blue right here that I'm tracing. These are called terminal cisterns or terminal cisternae. Terminal cisterns are just very, very enlarged, very swollen areas of a sarcoplasmic reticulum. Now, the sarcoplasmic reticulum has the same jobs as a smooth endoplasmic reticulum. Think of what those were. Well, the one that we're concerned with most in skeletal muscle or in muscle in general is the SR holds calcium. Because remember, if calcium is inside of a cell, it likes to attach to stuff. And in general, we don't want that to happen. So there's a lot of calcium inside of a muscle, but it's all stored inside the sarcoplasmic reticulum. Since the terminal cisterns are so enlarged, there's even more calcium inside of those. Now, it might be kind of hard to see in this image here, but remember this purple tube right here? This was a transverse tubule or a T tubule. Look closely at that. On each side of it, we see a terminal cistern. And they are actually joined together by some very specialized proteins we'll learn about in the next lecture. One T tubule and both of its adjacent terminal cisternae. So one T tubule and the two terminal cisternae that are right next to it, we call that a triad. A triad is one T tubule and the two terminal cisternae that bump up against it. That will be very important in our next lecture. Now, inside of the sarcoplasmic reticulum, there's a special protein called calsequestrin. Calsequestrin is a protein that really likes calcium. And what its job is to pull calcium into the sarcoplasmic reticulum. And when the calcium comes in, it holds the calcium. So that is why there is so much calcium stored inside the sarcoplasmic reticulum. The calsequestrin pulls it in and it holds on to it once it's inside. Now going even smaller. We were already pretty small since we were looking inside of a muscle cell, but now we're going even smaller and we're going to be looking at those myofibrils. Let's go back here. So we've got this long myofibril right here, and I said that this was a bundle of some protein fibers. If we look at the picture close enough, we can see these kind of purplish areas right here, and these kind of black zigzag areas right here, and maybe some red striped or pink striped areas right here. Well, those stripes are actually protein strands, and that's what we're going to be looking at. We're going to be looking at the proteins that make up these myofibrils. when we look at those myofibrils, and that's kind of what we see down here at the bottom, we've just taken one area of this and enlarged it. And that's what we see down here at the bottom. We see these very, very large purple proteins and these smaller red and white proteins. And what we're looking at are called myofilaments. Myofilaments are these smaller protein strands that bundled together and made those myofibrils that we just looked at. So, myofilaments, and when we look at myofilaments, there's different strands of proteins that make up these myofilaments. Primarily, we're going to talk about thick filaments and thin filaments. Now, there are some others that go into it, but right now we're just going to be talking about the thick filaments and the thin filaments. First, let's look at the thick filaments. That's these purple proteins here. And that's what's enlarged up here in the top view. A thick filament is made of the protein myosin. Myosin is a protein, and it's actually going to have several proteins joined together to make the thick filament. What did we just learn that was called? It is a protein with quaternary structure. So let's look at the protein myosin. Myosin is a weird shaped protein in that remember when we talked about globular proteins and filamentous proteins? Myosin has both. Myosin has this area down here that's globular. It's called the head. And then it's got these long filaments, which is the tail. And we have two of these proteins side by side, and the tails wrap around each other. So there is our quaternary structure. And we take that quaternary structure even further by taking several of these and joining them together into this thick filament. So each of these heads is actually two myosin heads, and then we can see the tails are all laying parallel to each other. That is a thick filament. And in a muscle cell, we have this long, thick filament with heads down here on one end and the tails protruding this way. At the other end, we have several heads and the tails protruding this way. So we actually have two clusters of myosin on each of these thick filaments. The heads on one end with the tails pointing towards the middle, the heads on the other end also with the tails pointing towards the middle, and then right in the middle here we don't see any heads at all. This is called the bare zone. But this entire thing is a thick filament. Next, if we look at these red and white guys here, these are thin filaments. Thin filaments are actually made up of a few proteins, but primarily it's made of a protein called actin. Actin is a globular protein. We talk about G-actin. G-actin are the globular protein subunits. And that's what we see right down here, these guys that look like a blueberry that somebody stuck their thumb in or something and made a little dent. Over here they're blue, in this picture they're red. I just have the blue picture here to show you the actual individual subunits. And all of these globular subunits, all of these G-actin pieces join together to make long strands or long fibers of actin. That's these blue oval shaped things here. That is fibrous actin or F-actin. Over here in this image, it's the red guys. Over here in this image, it's the blue guys. But in either case, the individual ovals are G-actin, globular actin. This is the protein actin. But they join together to make a quaternary structure fiber called F-actin, fibrous actin. Now, when they're down here, we can see that there is this dipped in area. I said it looked like somebody stuck their thumb in it. Well, that's called the active site. This is what some certain things that we will see later like to attach to. But when it is in this fibrous form, we can see that there's actually a string, it's another protein, that's laying, covering up that active site. This protein, this long fiber that covers up the active site, is called tropomyosin. In this image, it's kind of an orange color. In this image, it's the white. But tropomyosin wraps around F-actin and covers up the active site on the actin. And to hold that tropomyosin in place, there are some anchoring proteins here called troponin. Troponin holds the tropomyosin in place so that the tropomyosin covers the active site on the actin. Now, there are some other proteins involved. So let's see what those other proteins are. First, there are several different types of elastic filaments. The ones that we're going to be concerned with are called titin. Titin are like little springs, and they grab onto the thick filaments and anchor the thick filaments in place. They're joining this guy right down here that we will see in just a little bit, but they're holding the thick filaments in place like a spring. There are also several different types of regulatory proteins. The one that we're going to be concerned with is called dystrophin. Dystrophin helps anchor the thick and thin filaments, really it anchors the myofibrils, to the sarcolemma, the plasma membrane of a muscle cell, and to the endomysium. The endomysium, remember, was the inner membrane that kind of wrapped around individual muscle cells. Now there's one more term here that I want to introduce, and that's called a cross bridge. So I'm going to go back to our previous image. Myosin heads really like these active sites on the actin. If the active sites on the actin were not covered up, the myosin heads would actually raise up and attach to them. If that were to happen, if the myosin and the actin joined together, that's called a cross bridge. But that does not typically happen in a muscle cell at rest because these active sites are covered up by the protein tropomyosin. If tropomyosin weren't there and these active sites were exposed, then the myosin would raise up and grab hold of the actin. So now when we look at a skeletal muscle cell under a microscope, which is what we see right here, it is striped. It looks stripy. So we say that it is striated. And we saw this back in the tissue chapter. Skeletal muscle and cardiac muscle were both striated. Well, striated just means that under a microscope, it looks striped. And that's because of the way that all of those different proteins are laid out. So down here at the bottom, we see how those proteins are laid out. Up here at the top, we see that it repeats one after another, after another, after another. So what we see right down here is one group of those proteins. But off to this side and off to this side, we see a little bit of that same layout repeating. Up here, the same thing. Here's one, and then it repeats, and then it would repeat, and it would repeat, and it would repeat, and it would repeat. The length of the myofibril and the length of the muscle cell and the length of the whole muscle. So we have these repeating units, and the unit we can kind of look at from this zigzag area right here to this zigzag area right here. I'll kind of draw it out. So this zigzaggy area right here and this zigzaggy area right here. Just for ease of describing the different parts, we're going to look at each of these two zigzaggy areas, and we're going to call that a Z-disc. The area in between the two Z-discs is called a sarcomere. So that's what we see right down here. From this Z-disc to this Z-disc, everything in between, this is called a sarcomere. And a sarcomere is defined as the contractile unit of muscle. The contractile unit of muscle. And when we look at a sarcomere, based on which proteins are in a particular area, we can name different parts of this sarcomere by different names. First, look at the thick filaments, this purple guy right here. From one end of the thick filament all the way to the other end of a thick filament is called the A band. The A band is the length of a thick filament of one sarcomere. Now, as we just saw, a sarcomere runs from Z-disc to Z-disc. So between the Z-disc and where the thick filament starts, there's an area where there is no thick filament. Same thing on the other side of the Z-disc. There's an area where there are no thick filaments. So the area in between the thick filaments, the area in between the A band of one sarcomere and the thick filaments, or the A band of the neighboring sarcomere, is called the I band. The I band is the area in between the thick filaments, or the A bands, of one sarcomere and the neighboring sarcomere. That's the I band. Now, if we look at the Z-discs, here we can see the green guys. This is that titin, the elastic filament that we mentioned. So the titin, or the elastic filament, is anchoring the thick filament to the Z-disc. But also coming off of the Z-disc are the thin filaments. So projecting from this Z-disc towards the middle of this sarcomere are thin filaments. If we look at the other Z-disc, projecting from this Z-disc towards the middle of this sarcomere are more thin filaments. Now, the area in between the thin filaments from one Z-disc and the thin filaments from another Z-disc, this is called the H-band. The H-band, we can talk about it a couple of different ways. We can say that the H-band is the area in between the thin filaments from one Z-disc and the thin filaments from another Z-disc. Or we can say that the H-band is the area where there is only thick filament. There is only thick filament in the H-band. Now, running right through the middle of the H-band is a protein that's going perpendicular to all of the other proteins that we've talked about so far. This is called the M line. I think of it as the mid line of the sarcomere. But the M line is a protein that all of the thick filaments of this sarcomere are anchored to. It kind of holds these thick filaments in place, the M line. And finally, re-mentioning the Z-disc. The Z-disc is the starting and ending point of a sarcomere. So the sarcomere is everything in between two adjacent Z-discs. The Z-discs are also what the thin filaments are attached to, and the Z-disc is what the titin is attached to on one side, and the titin is attached to the thick filaments on the other side. So that's the anatomy of muscle, the microanatomy of muscle. Now, next 2 lectures, we're going to be looking at how muscle functions. Please make sure everything that we talked about today makes sense, because when we start talking about muscle contractions, we're going to be referring to these different proteins, and we're going to be referring to these different parts of the sarcomere. So if you have any questions at all, ask me, ask your tutor, ask a lab instructor, but make sure to get any questions that you have cleared up. All right, take care, and I will talk to you next time.